we look at soil health, there's a lot of different definitions of it, but Dr. Kremer cloned this in 2016. Soil health focuses on the living, dynamic nature of the soil that functions by incorporating the biological attributes of the biodiversity, food web structure, ecosystem functioning, and intimate relationship of soil microorganisms of plants and animals. Say that fast. So the main functions of soils are organic matter formation, water infiltration, filtration and storage, atmospheric modification, habitat for microorganisms, and a medium for plant growth. Now this is an example of this was a test done in biological activity of different microbes within an orchard system. Now if we look at, this was an organic orchard, and all these different colors represent different bacteria and fungi that are present in that soil system. This was an organic orchard. Here, so you can see that's pretty high. Here's an organic orchard that had some biological additives added to it. And if we look at a cultivated system that a lot of us are in with cropping, you only have about half the biology that the organic system actually has. We look at a no-till system. We're kind of like this resting prairie here a little bit, and a strip till is more like this grass system here. So all those models have more biological activity than a cultivated system. So cultivation destroys that house with its bacteria and fungi. If we look at this, you've seen this probably before, is the important aspects of soil structure. We got the physical, the chemical, and biological. This is really where the soil health is, where they overlap. If we look at the physical characteristics of the soil, we're looking at the bulk density, the aggregate stability, pore size distribution, that water-filled pore space was talked about earlier, water holding capacity, water infiltration rate, soil compaction, topsoil depth. If we look at the biological aspects, we're looking at the microbial biomass, getting a little dry mouth here. Microbial community, structure, biodiversity, microbial activity, soil enzyme activity, and soil respiration. Potentially mineralizable and soil dispersive, disease suppression, nematode assessment, earthworm assessment, glomalin content. So a lot of these we're talking about what we measure, again in soil health as well. We look at the chemical aspect of it. We're looking at soil organic matter, the active carbon, soil pH, electrical conductivity, cation exchange capacity, available phosphorus, exchange with potassium, and the sodium absorption rate. The main threats to our soil, certainly we've seen a lot of soil erosion. We've seen organic carbon loss. We have nutrient imbalances, compaction, the loss of biodiversity, soil salinification, solidification, acidification, soil contamination, and soil sealing. If we look at soil erosion, we look at the accelerated removal of topsoil from the land surface through water, wind, or tillage. Soil compaction, increase in density and decline of macroporosity in soil that impairs soil function and impedes root penetration, water and gas exchange. Soil sealing is permanent covering of the soil surface with impermeable artificial materials. As our cities grow, we pour more concrete roads. That's sealing those soils off permanently. Wind erosion. Took this picture going down the road. You can't tell where the field ends and the road ditch starts. A lot of dirt blowing across there. For every 1 ounce per square foot of soil that's lost, that's almost 1.5 tons per acre of soil that you lost off your field. That 1 ounce in 1 square foot is not very big. So that's why it's really important to stabilize our soils. Water erosion, we can certainly have a lot of runoff. This is after a heavy rain. Good soil structure over here. You didn't see a water ponding or runoff here. But again, so it just shows the difference improvement in soil structure can make to handling a heavy load of rain. Compaction, we certainly dealt with this last year. Low-lying areas, even some better ground, a lot of moisture. We still got in when we had to, so we compacted that soil. This is actually a corn root mass here that you look at that and say, well, that's a lot of root there. But that was in soil that was heavily compacted. Those roots are all only about a root, inch and a half deep. You couldn't go any further. The ground was that compacted and they all went sideways. So management keys for reducing erosion, compaction, soil sealing, reduce that soil disturbance, increase plant diversity, certainly rotation, perennial crops, cover crops can factor into that, continuous living roots in soils as long as we possibly can, keep soil surface covered at all times, build our soil organic matter, which is going to be our crop residue management, manure, compost, mulch, biological products, and cover crops. And we want to put an integrated and diversified management system in place. Organic matter, I just talked about earlier, so you can see we're driving home some of the same factors. So we're looking at supply. Organic matter supplies many nutrients, including the nitrogen, phosphorus, sulfur, and trace minerals. It improves the soil structure, aeration and drainage, can reduce that erosion and increase CEC. Also feeds the beneficial soil life, microbes and earthworms. For every 1% increase in organic matter, available nitrogen increases 10 to 20 pounds per acre. For every 1% increase in organic matter, soil watering and holding capacity goes up as well. So important biological attributes of healing soils are certainly influenced by the crops that are grown on them. Here's a cover crop of really good looking red clover, which will provide carbon and nitrogen in the soils. We look at what's below the surface. Look, these are roots here. These are rhizobacteria on the root surface that are colonizing, interacting with the plant. We need to kind of know what's below us. So this is important. How many of you have livestock at home? Okay. The rest of you have livestock under the soil. There are more microbes in one handful of soil than there are people on the earth. There's 4,000, 8,000 pounds of microbes per acre in the top 6 inches of soil. So we look at the soil structure, and again, we heard a little bit about that earlier too, is we look at how those particles are all put together. Soil, sand, silt, organic matter, and clay particles can be attached to one another. Whoops, excuse me. Or they can be clumped together. So if we got them clumped together into aggregates, we've got what we refer to as flocculated soil. That's going to give us the opportunity to have some air and water space in there. See, we got this flocculated soil has air space in there. Get this clumped together with compacted, tight soils like that clay, high magnesium soil we saw earlier. We have that flocculated soil. We certainly have room for water to penetrate that. Roots penetrate that as well. So most organic matter and clay particles have negative electrical charge. Charges repel, so these particles resist attaching to one another. This can result in dispersed or unaggregated soil structure. Cations, a positively charged atom or molecule. Common soil cations include sodium, potassium, magnesium, ammonium, and calcium. The absorbed cations can make soil particles clump together or flocculate, again to create that aggregated soil structure. so we have better water holding capacity. Those nutrients can penetrate, get up into the roots. Roots can actually go down, get more nutrition. This is an example of dispersed or flocculated soils. Like we saw earlier in the slide, this is after a hard rain. That water is all standing here. This is tight soil, compacted, dispersed. We're going to look at the flocculated soil here. At that same hard rain, there's no water standing. That soil had place for that water to go to, that water holding capacity. To look at loss of diversity in soil contamination, the loss of soil biodiversity is decline in diversity of organisms present in soil that affect multiple ecosystem functions, including plant diversity, decomposition, nutrient retention and cycling, plant and animal health, soil carbon sequestration, and greenhouse gas emissions. Well, soil contamination is increase in toxic compounds or heavy metals or pesticides, and they affect the human health and provision of our soil ecosystem. Three major pathways for diffusing soil contamination are atmospheric, agriculture, and flood events. Soil contamination can reduce food security by decreasing crop yields and rendering crops unsafe for consumption. A lot of this is, the food health is driven by our consumers, and as food growers, we need to recognize that and address that. We look at the plants and look at the biology. These plants talk to one another, they supply nutrients to each other. This is actually corn root that we actually have droplets here at the end. That's actually sugar droplets feeding the biology in the soil. A lot of different things going on in the soil. We've got our bacteria, we've got our fungi, we've got algae, earthworms, nematodes. There's certainly viruses there as well. We like to dig up that soil and see what we have. Earthworms are the macro part of the biology. That's what we can see. So from an on-farm test, that's a good thing to do, is go up and dig up some dirt, see what you have for earthworm population there. That we can see. The rest of these we're going to have to look at a microscope to see. There's a lot of different things in that soil. Think about 3 miles of fungi. Fungi are thousandths of an inch in diameter, so very, very small, but they can grow to be 3 yards long. So you think about tillage going through the field, you can tear up a lot of those. So again, the livestock below the soil surface. We look at traditional management prescribed to adjust soil biology and function versus a cover crop. We had a simple cover crop in, and we look at our atmospheric mycorrhizal fungi population. We had twice what we did without having any cover crop. And those actually do get into the plant and can help mobilize nutrients, particularly phosphorus. This slide is really hard to see, but really what it's saying is, and I apologize for that, is that we have a lot of different chemicals that actually go into the soil. We're applying to the soil and how they affect the different nutrients in the soil and biology. Soil contamination. Did you know that these nodes in a corn plant also are your filters for that corn plant? They actually will filter out bad nutrients, try to reduce pesticide going up into the plant. So we split those plants and look at those. You can see that there's a lot of clouding, darkness here. Those filters are getting plugged, so it's hard for other nutrients to get up and down that plant. These plants have a lot less of that. This farm had split this field and kept farming one conventionally and one biologically. You can see the difference in corn. Soil organic carbon loss. Decline of organic carbon stock in the soil affects its fertility status, and climate change regulation capacity. Primary driver of soil organic carbon is lost globally as land use change and subsequent management practices. Over the years, we've changed a lot from how we manage our soil versus what our forefathers did. Now, the last few years has been a lot more emphasis put on soil health and micronutrients, which is a good thing. Net gain or loss of nutrients from the soil zone that is accessible to plant roots is the incorrect land use and management result in excess of nutrient-causing soil contamination. So we look at that organic matter. See, we've got some fresh residue that will hopefully convert to our organic matter. We've got our decomposing organic matter. We've got our bacteria and fungi that are working on that. That's about anywhere from 33 to 50% of that total organic matter. We've got stabilized organic matter. Again, that can be in that 33 to 50% range. More is absolutely better because that's our humus. And then we've got our living organisms here. So again, a lot of things are taking place to create that soil organic matter. The plant tissue's got to break down the bacteria and fungi, creating that soil organic matter and putting off CO2. We look at different types of nutrients and how they affect turnover time in that soil organic matter. We've got our root amino acids and sugars here. Here's a timeline on the bottom. You can see that they're going to break down in hours and days. Sugars are pretty fast. Our unsaturated hydrocarbons are fast. Carbohydrates, depending upon what they are, can take many years. Lignin, or tannin, cellulose, undigestible fibers, again, can take years to break down, depending upon the type that they are. Some will break down in days, but it takes a long time to change over that organic matter. We look at inorganic fertilizers versus organic materials. We certainly see that we have a negative effect in that soil biology. We're going to deplete our organic matter in that soil. We reduce our fungi population. We have nutrient losses. If we're using organic materials, we can trigger that release of phosphorus, sulfur, and potassium. We can trigger our growth regulators. We can mobilize our trace minerals. We can suppress our pathogens. We have better stress tolerance for the types of cropping we've had the last couple of years. Ultimately, our goal is to improve that soil health. And that's why Mike and Andrea have partnered with the people that they have, is that they are concerned about that soil health and soil biology and raising those better crops. And this system certainly does that. We look at chemical properties. We look at our chemicals that are out there. We certainly know that we've got some problems with nutrient tie-up. Dicamba ties up copper. We know glyphosate is going to tie up our magnesium, copper, zinc. So again, I apologize, the slide's kind of hard to see, but it has different chemicals on there and tie-ups of the trace minerals that they have. We look at trace minerals and micronutrient stressors. Micronutrients are the most immobile nutrients. Therefore, anything that inhibits new root growth will inhibit uptake of those nutrients. Acid soils will increase uptake, and high pH soils will inhibit the uptake of these trace minerals. Micronutrients are readily and tightly complexed by organic matter. Therefore, higher soil organic matter levels will reduce the availability of these trace minerals. And waterlogged soils will certainly reduce the micro content and availability while they're saturated. However, after they're drained, the nutrients become available again. So again, looking at that soil structure, improving that soil structure is going to make nutrients more available to the plant as well. And of course, we have our chelation from application of chemicals. We look at those functional trace minerals. We look at copper. It's a catalyst in photosynthesis and respiration. So again, important for plant function. It's important for several enzyme systems involved in building and converting amino acids to protein. So we're looking at protein, we're looking at our feedstuffs, and we're short in protein. Might want to certainly look at where our copper levels are at. It's important in carbohydrate and protein metabolism. It assists in the formation of lignin in the plant cell walls, so it gives that plant strength and standability. Copper can also affect the flavor, the storage ability, and the sugar content of fruits. Zinc, production of auxins, an essential growth hormone. It activates enzymes in protein synthesis. That's involved in regulation and consumption of sugars. It's necessary for starch formation and proper root development. Zinc influences the rate of seed and stalk maturity. Again, that's important. Necessary for the formation of chlorophyll and carbohydrates. The presence of adequate amounts in this tissue enables a plant to withstand lower air temperatures. So you can see as we talk about these trace minerals, for some of the same reasons they're important in plant health, they're important for animal health, they're important for human health. Manganese, stimulation of carbon dioxide in photosynthesis. It aids in the synthesis of chlorophyll and nitrate assimilation. Activates fat-forming enzymes. Functions in the formation of riboflavin, ascorbic acid, and carotene. Functions in electron transport during photosynthesis. It's important in the Hill reaction. Water is split during photosynthesis. Boron, maintaining a balance between sugar and starch. Translocation of sugar and carbohydrates. So boron as a nutrient doesn't translocate up and down the plant, but it does trigger sugar translocation. It's important in pollination and seed production. Necessary for normal cell division, nitrogen metabolism, and protein formation. It's essential for proper cell wall formation. Plays an important role in the proper function of cell membranes and the transportation of potassium to guard cells. Proper control of internal water balances. Remember, potassium is an electrolyte. Sulfur, not certainly a trace mineral, but again, a very important secondary nutrient. This is something you want to be certainly looking at and addressing every year as well. When we'll talk about looking at nitrogen to sulfur ratio in plants, we'll talk about a 10-to-1 nitrogen to sulfur ratio being important because pests, insects can't digest complete proteins. So we get that nitrogen to sulfur ratio balanced out 10-to-1. You've got natural insect resistance in that plant as well. An example of one of our products is a test, which is a copper product. So looking at application of that 30 to 40 days after planting with your first post-herbicide application, applying 8 ounces of a test for plant health, plant cell wall structure and strength. And again, approximately 30 days later at flag leaf emergence. Apply that with a fungicide application. Again, 8 ounces for plant health and building, converting amino acids and protein. Again, this is a copper. So we're going to look at getting that into that plant and creating more amino acids and proteins. Additional cost is approximately $16 per acre. Payback is in plant health and in yield. We can do this in dry soybeans and dry beans as well at B2 and B4. Again, with the first post-herbicide application. Again, we're talking about plant health, applying at R1 to R2 with the fungicide application. Nanotechnology. One of the things that we do with ST Biologicals, the ST stands for Source Technology. And so one of the things we do is we source other technology that will be beneficial to you, the producer. We screen those products, go through them, determine what is the best product to bring forth to the marketplace. So nanotechnology is used in a lot of different things. Actually, scratch-proofing your eyeglasses, crack-resistant paints, so there's one-coat paints. We use nanotechnology. So nanograffiti coatings, transparent sunscreens, stain-repellent fabrics, ceramic coating for solar cells, electronics. So a calcium ion, we're talking about the nanotechnology, we supply anions to the plant. And so we look at a calcium ion, to give you an idea of size, that's 1,000 of a nanoparticle. You put 1,000 calcium particles into 1 nanoparticle. You put 1,000 nanoparticles into a plant cell, and you put 1,000 plant cells into a poppy seed. Gives you an idea how small these ions actually are. We can look at, they're small enough to cross the biological barriers, and they're large enough to carry a significant payload. So going back to looking at like a wiffle ball, if those nanoparticles have holes in them, they have electrical charges, positive or negative, and we can actually load those with specific nutrients and deliver those to the plant. They can actually get through that outer layer of that plant very easily. Here's a microscopic picture of that nanoparticle. See, it's got the holes in there, but under higher magnification, you see it's got like little pins on it as well. So when this is actually sprayed onto the plants, it sticks on the plant, it stays with the plant. It helps translocate those nutrients into that plant then. So we can get through that plant leaf. You get down through, so you've got guard cells there, can get down through the guard cells. It will stay attached. Rain will not wash these nanoparticles off. Get through cracks, any cracks in that leaf layer. Another product we have is calcine. This is for using it for improving soil structure. We can certainly change that soil structure with the use of calcium and water in this calcine product. We've had a lot of great results with this. We started with this out in Arizona. We've used it out in the Dakotas pretty heavily, and now in Minnesota, Wisconsin. It will open up those tight, poorly drained soils. Top 5 mistakes in soil application is the wrong products used, or the wrong amount, wrong place, wrong time, not following a consistent program. When working with who's ever consulting your farm, it's important to find out what your limiting factors are, to target those, and work to eliminate those. Products coming, we've got some new products coming down the line. In the nanotechnology, we're going to have a, hopefully, don't know if it'll be out yet this spring or not, but it's a slow release nitrogen product. We'll have some products in the STB line that be organic, so they test and instill the organic versions. We're only a couple of weeks away from having those. A couple little sayings here. You fail, never give up, because fail stands for first attempt in learning. End is not the end. In fact, END stands for effort never dies. If you get no, NO means next opportunity. I thank you for your attention, and Don and I'll be around this afternoon to answer any questions as well. We have a lot of products that we can talk about, too numerous to put up and take this time now, but we can certainly talk about those later. Thank you.